Hi, everybody. I'm Satish. Thanks very much to the conference organizers for giving me the opportunity to talk today. That's me with the completely meaningless graphic up here. So actually, both my graphic as well as my title are really meaningless because I wanted to have the most non-committal title so I could really decide what I could speak about last night. But hopefully there'll be some correspondence between what I'm actually discussing here. Oh no, my wife's here. All right, now I got to kick it up a notch. Okay, so I'm guessing a lot of you are familiar with this. This is the basic natural history of HIV infection in the modern era of antiretroviral therapy. And so what you can see is initially there's a period of viremic disease. And when an individual goes on antiretroviral therapy, the viral load decays very rapidly to below clinically detectable levels. And with the modern regimens, really, the virus typically stays at undetectable levels. But the second that well, I shouldn't say the second, but when antiretroviral therapy is stopped, the virus almost universally rebounds. And in most cases, it rebounds within just a few weeks. So why is that the case? So the simple explanation is that antiretroviral therapy blocks viral replication, but it doesn't do anything to destroy any of the infected cells that pre-existed the onset of antiretroviral therapy. So to explain this extremely simply, if we look at the setting of untreated HIV disease, we can think of the process as follows. You have HIV-infected cells. They'll produce virions. They'll produce viral particles. Those viruses will go on to encounter uninfected cells and create a new generation of infected cells. And of course, this goes on ad infinitum. But in the setting of treated HIV infection, essentially that whole right side of the schematic is taken off the radar screen. So there's no more new replication or no de novo infection. But what you do have are these HIV-infected cells, which are often these long-lived, latently infected central memory CD4 T cells. And they tend to be invisible to the immune system. And there's at least some evidence suggesting that these cells can persist for months or even years. And so, as Paul mentioned briefly, the field is really, really focused on this idea of being able to eradicate the latent reservoir to achieve a cure for HIV infection. So another detail that's become more clear in the last few years is that the size of this latent HIV reservoir determines viral rebound kinetics following the cessation of antiretroviral therapy. So what you can see is there's very considerable variability in the size of the latent reservoir across infected individuals. And I'm going to show you some data from our group that adds some data to that concept. So this figure right here is from a review written by Dr. Stephen Deeks, who presented to all of you here last year. So what you can see, the typical rebound time for individuals who stop antiretroviral therapy is within a couple of months. But then there were these really fascinating cases that have received a lot of media attention, including the cases of the Boston patients, as well as the Mississippi baby, as well as the infamous Berlin patient, or Timothy Ray Brown. And these are individuals who have a smaller than normal latent reservoir. And there have been a few different mathematical modeling efforts that have shown that the size of the latent reservoir, or the assumed size of the latent reservoir in these individuals, largely and relatively accurately predicts the time until the virus rebounds after stopping antiretroviral therapy. So really the central thesis of the talk that I'm going to give today and the data that I'm going to show you is this, that if we can identify host factors that naturally modulate HIV reservoir size in vivo, this will help us to cure HIV infection. And there's a couple of different ways that these data will help. So if we can identify host factors that modulate the HIV reservoir, those should serve as new pharmacologic targets that we can hit using a variety of different strategies. In addition to pharmacologic targets, if we can identify host factors that regulate the size of the reservoir, we may also be able to leverage these markers as clinically useful biomarkers within cure trials. So I don't have the time to go into it today, but one of the key challenges associated with the HIV cure field is we don't really have any useful or very useful clinical indicators of efficacy when we've attempted some sort of curative intervention in the clinic. So really what the field is looking for now is something like an HIV viral load that we have for managing patients with antiretroviral therapy. So basically what I'm going to talk to you about today, we've employed this highly translational systems biology approach to identify modulators and markers of the latent HIV reservoir. And this is a schematic of our sampling strategy. So we assembled a cohort of 72 HIV-infected individuals on antiretroviral therapy, and all of them were on antiretroviral therapy for between 1 to 2 years. We isolated blood plasma, and then we measured we measured viral load using an ultrasensitive assay. So one key challenge, and again, I don't have that much time to go into it, but how we even measure the latent reservoir is incredibly challenging, and there's no real field consensus on how we should measure it. There are a few different ways that seem intuitive that are generally accepted. One is looking at residual viremia using an ultrasensitive assay. Another two strategies that we used, we isolated CD4 T cells from peripheral blood. We extracted both RNA and DNA. And then we used quantitative PCR to measure cell-associated HIV RNA. So we looked at the frequency of HIV transcripts, essentially, in a population of CD4 T cells. The other thing that we did is we looked at the frequency of HIV proviral DNA. So we looked at both the HIV Pol gene and frequencies of 2-LTR circles, which are often thought to be indicators of abortive HIV integration. So this is the assortment of HIV reservoir measurements that we performed. So in terms of the correlates that we're interested in, we did a whole range of immunophenotyping to look at the surface expression of receptors and co-receptors that are associated with HIV, as well as a range of activation and exhaustion markers that are indirectly correlated with HIV persistence. Then within the plasma compartment, we measured anti-HIV antibodies. We did global cytokine profiling. We investigated these things called exosomes, or extracellular vesicles, which are these key intermediates in intercellular communication. We did two different transcriptomic steps. So we did global transcriptomics using RNA sequencing, and then we also did confirmatory analyses of targeted factors, specifically host restriction factors that I'll talk about briefly. And then on the genomic DNA front, we did some limited host genotyping. So in particular, we looked for SNPs that were associated with HIV disease progression outside of the context of antiretroviral treatment. And we wanted to know if those same SNPs had prognostic value within the setting of treated disease. And then we also did ultra-deep sequencing of HIV DNA to see if there were any correlations between the population genetics of HIV and the overall viral burden. But the only thing I'm going to talk about today, I'm going to just walk through one vignette out of this kind of systems biology approach, where we were able to go from an in vivo transcriptome to a putative mechanism to a putative novel therapeutic approach to eradicate the HIV reservoir. So I mentioned that we were particularly interested in host restriction factors. So what are host restriction factors? Restriction factors are, I think, can be very briefly described as a suite of proteins that attack HIV directly within the target cell where it makes a living. So a lot of the gene expression data that I'm going to discuss are relevant to the expression levels of these factors that attack HIV right inside the target cell. So the first thing that we did was we measured HIV reservoir size using those three different methods that I discussed. And there are a few take-home messages here. One is that there's multi-log variability in everything that we measured. So if we look at the frequency of HIV RNA or the frequency of HIV DNA, and again, we have two different forms of HIV DNA, in all patients, you can see that there's 3 to 4 log variability in the size of their HIV reservoir. The other thing that was very consistent was that the timing of antiretroviral therapy initiation had a profound effect on the size of the reservoir. So individuals on the right side of these graphs started treatment after a year into infection, and the individuals on the left side of each of these graphs started treatment within about 6 months of infection. And this has been reported by multiple groups at this point, but we see a very consistent effect of early antiretroviral therapy. So the main question we wanted to address was, could we identify particular genes using both RNA sequencing as well as a targeted quantitative PCR approach? Could we find any genes whose expression levels correlated very significantly with the size of the latent reservoir? And again, we used very stringent statistical methods, in this case a Bonferroni correction, to correct for multiple comparisons. And what you can see here, the two genes that were most significantly correlated with HIV transcription were P21 and schlafen-11. So these are two established antiviral host restriction factors that have activity against HIV, and they also happen to be well-established cell cycle regulators. So if you look at these graphs, what you can see here is the more P21 expression you have, the lower the frequency of HIV RNA and CD4 T cells. And the relationship is very similar for schlafen-11. The more schlafen-11 expression you have in a CD4 T cell, the less HIV RNA you encounter in those cells. And so this certainly does not prove causality in any way, but we interpreted these data to suggest that we might be honing in on some sort of causal relationship, that these factors may actually be suppressing HIV transcription in the setting of antiretroviral therapy. So since it seemed like these things could be regulators of HIV transcription, we immediately drew the connection between these relationships that we were tuning into and this shock and kill HIV eradication approach, which I'm guessing many of you are familiar with. So the shock and kill HIV eradication strategy is conceived as follows. So we take latently infected cells, we administer some sort of shock agent that will reactivate the latent virus within those cells. All of this will be done during antiretroviral therapy, because the last thing we'd want to do is reactivate a bunch of viruses that would go on to infect new cells. But hopefully in the setting of antiretroviral therapy, the virus would be released, but you'd have no new infection events. And there's a huge leap of faith with this side of the equation, but what we hope is that by reactivating latent virus within these cells, the cells themselves would die. And so one way that they would die is through direct cytopathic effects that are mediated by the viral proteins, which are likely toxic. And another thing that we hope will happen is that you'll have presentation of viral antigen, so cells like CD8 cells can come along and destroy the infected cell. To date, I don't think anybody would argue with me about this. I think the success associated with this approach in the clinic, there's been about 9 or 10 different clinical studies, and the success has been meager at best. And one of the reasons is that the shock agents that we've used are not very potent. So I think the field, if we're going to pursue the strategy, the field desperately needs more powerful shocking approaches. So we surfed through the literature to find that galectin-9, a sugar-binding protein, is an upstream regulator of P21. So we were looking for some way that we could manipulate P21 expression to see if it would indirectly affect HIV transcription. And so the hypothesis we pursued is that galectin-9 promotes the reversal of HIV latency through its regulatory interaction with P21. And those are the data I'll present to you now. So the first thing that we did is we measured the effects of galectin-9 administration, which again is this carbohydrate-binding protein. It's a lectin. We measured the effects of galectin-9 administration on the reactivation of latent HIV in an in vitro model called the J-Lat cell. So the J-Lat model was invented by Eric Verdin at the Gladstone. And basically they took Jurkat cells, they took a Jurkat CD4 T cell line, and they stably infected them with a provirus. So they're all latently infected with a single copy of the virus. And what you can see here is when we treat the cells with CD3, CD28, or TNF-alpha, or PMA ionomycin, which we know are established reactivators of latent virus, we can see this huge GFP signal, since the latent virus in these cells expresses GFP. We tried a whole bunch of other galectin family members, like galectin 1, 3, 4, 7, and 8, and they didn't do anything. So we saw no evidence of any viral reactivation using these other galectin members. When we administered galectin-9, we actually see a dose-dependent induction of GFP expression, which indicates that the virus is being reactivated. So this looked promising, but again, as we all know, cell lines don't always behave like actual real cells. So the next thing we did was a real experiment where we did large volume blood draws from 13 individuals who were infected with HIV, who were on antiretroviral therapy. And we tested the capacity of this recombinant galectin-9 protein to reactivate latent virus in the patient-derived cells ex vivo. And what we can see really reinforces what we observed in our in vitro experiment. So vorinostat is an established latency reversal agent. You can see that we get a moderate reactivation of virus in response to vorinostat. Both dosages of galectin-9 that we used reactivated virus significantly more potently than vorinostat or our DMSO control. And of course, PMA ionomycin was the most powerful, but you can't really go into human beings with PMA ionomycin, I don't think. So the next question we had was, okay, we were pretty convinced that this was a real result. And one thing that I don't have the time to present here, but is in this paper in PLOS Pathogens, we did a lot of characterization on how galectin-9 affects the viability of cells, proliferation, the activation state, et cetera. So we thought we had something interesting. The next thing we wanted to do was to dig into the actual mechanism. And if you look through the literature, there are three main receptors that are associated with galectin-9. There's TIM3, PDI, which stands for protein disulfide isomerase, and then CD44. Long story short, when we competitively bound these receptors with antibodies, it in no way affected the Gal9-mediated reactivation of HIV, suggesting that none of these canonical receptors that are reported in the literature are the key intermediates in galectin-9 signaling, at least in this context. So Mohammed, who led all of this work, he came up with the idea that since this is a carbohydrate-binding protein, that a reasonable hypothesis would be that carbohydrates at the surface of the CD4 T cell would play a very critical role in the signaling. And so he did a series of experiments where he deglycosylated these cells. He used a deglycosylation mix which stripped glycans off of the cell surface, and he also used tunicamycin, which completely shuts down the glycan synthesis process within the cell. And what you can see here is, so these agents, the deglycosylation agents, do not reactivate virus in any way, but you can see that there's a stepwise decay in the amount of reactivation you see when you administer the deglycosylation mix as well as tunicamycin. And data that I'm not showing here is that these deglycosylation procedures did nothing to affect reactivation by TNF-alpha or any of the other latency reversal agents. So this really suggested that glycans at the cell surface were playing a critical role in the signaling associated with this phenotype. The other thing that we noticed in our RNA sequencing data, so we actually generated RNA sequencing data from our in vitro experiments as well, and we could see that all of the ApoBec3 cytidine deaminase family members were very strongly induced by galectin-9, in contrast to vorinostat, which really didn't, which actually suppressed it to some degree. And the reason why we were excited about that piece of biology, so I actually pilfered this figure from Judd, who gave a talk earlier today from a paper that he wrote in J-Virology. So the way that the ApoBec3 family cytidine deaminases attack HIV, they're expressed in the producer cell, and when you have a virus emerging from the producer cell, it packages ApoBec within the emerging virion. Then when the virion goes to infect a new cell, during the reverse transcription process, the ApoBec3 enzyme plucks off the cytosines and replaces them with uracils. And so the complementary strand that gets integrated into the host genome is littered with G to A mutations, and it's no longer capable of encoding a viable gamut of proteins. And so this observation that ApoBec3 was induced was really interesting to us. And to take a closer look, we did a targeted qPCR just on the ApoBec3 gene, and this is in the the ex vivo data. You can see that there was a potent, like 10-fold induction of ApoBec3G expression in response to Gal9 in CD4 T cells, whereas vorinostat actually suppressed ApoBec3G expression. And interestingly, there are a couple of papers that report that vorinostat enhances the susceptibility of CD4 T cells to infection. Excuse me. And just to put this in perspective, our group has done some earlier work on interferon treatment in patients. And we could show that patients that underwent interferon treatment had kind of a meager, marginal, twofold elevation in the expression of their ApoBec3 family members. But then in another study that we reported that when we actually look at the population genetics of HIV within those individuals, you see lots of sequences on these long branch lengths, and these viruses are actually severely hypermutated, specifically during the interferon treatment period. So the take-home conclusion of this paper was that even a marginal induction of ApoBec3G in vivo can have a dramatic effect on the replication capacity of the virus in vivo. So we tested that in our culture system, and what we could see was, this is not a latency experiment, by the way. This is propagating infection. When we treated virus with PBS, obviously it didn't do anything. When we treated the cultures with interferon alpha, it significantly suppressed viral production, and Gal9 suppressed viral production even more. But what we really wanted to see was, on a per virus basis, we wanted to see if the infectivity of HIV was suppressed by galectin-9 treatment. And what we could see was, if we look on this right panel, this actually corrects for the P24 input. And if you correct for the amount of virus that's produced, viruses that were generated in the presence of Gal9 on average were about a log lower in their infectivity in CD4 T cells. So this really supports the idea that lethal mutagenesis at the hands of ApoBec3G in response to Gal9 induction decimates the infectivity of the virus. So to conclude, select host restriction factors likely modulate HIV persistence during antiretroviral therapy. This recombinant galectin-9 protein may promote eradication of the latent HIV reservoir within the shock and kill HIV cure framework. This RGAL9 mediated lethal mutagenesis of HIV may prove useful within the context of suboptimal antiretroviral therapy to prevent inadvertent reseeding of the viral reservoir when latency is reversed therapeutically. And I think this concern is becoming more relevant. I think there's more and more evidence that suggests that there's probably a little bit of residual ongoing viral replication during antiretroviral therapy, especially in tissues that are characterized by suboptimal penetration of some of these drugs. So I think I wouldn't feel safe assuming that 100% of infection would be blocked during a latency reversal approach. This approach would decimate the fitness of the virus as it exits the cell and would really serve as an insurance in that context. And in terms of ongoing work and future directions, right now, we've tested this in vitro and ex vivo. So we've already gone into a couple of different animal models. We've gone into humanized mice, and we've done some safety and tolerability experiments in the non-human primate model. But really, we're looking to see if any of the things that we've observed in a petri dish will extend to HIV infection in vivo. We have a new grant from NIMH to study the role of galectin-9 signaling in HIV persistence in the central nervous system. And this is probably maybe the most vigorous area in my lab right now. We're using a couple of different CRISPR-Cas9 approaches to both knock down and overexpress select host restriction factors in HIV latency models. Some of this work we're doing in collaboration with David Gordon and Nevin Krogan's lab, using a similar approach that Judd described, using the Cas9 ribonucleoprotein complex. And in our lab, we've set up the lentiviral system, where we're expressing Cas9 in a lentiviral construct. And just two quick acknowledgement slides. Firstly, I wanted to thank the amfAR in general, and also the amfAR Institute for HIV Cure Research. So a lot of the work that you saw, and definitely the downstream work that will come out of this, will be under the auspices of the amfAR Institute for HIV Cure Research that a group of us started here last year. And it's built off of a $20 million 5-year grant from amfAR. And in terms of giving credit to the people who did the work, Mohammed Abdel Mohsen is an absolutely fantastic scientist who did most of the work that I presented to you today. So he was a stellar postdoc in the lab who's now an assistant professor at the Wistar Institute. And actually, the work that he's building off of this is based on glycobiology. So he's really interested in how glycan interactions at the infected cell surface can be used to reverse HIV latency. And of course, I want to thank all my collaborators and all the participants in both the UCSF SCOPE cohort as well as the options cohort, and funding from the NIH and amfAR. And this is BSRI over here at the other side of the Golden Gate Bridge, so I hope that one day you guys stop by. Thank you.
Hi, do you know if other latency reversing agents also could be acting through the galectin-9 pathway or the mediators of the galectin-9 pathway?
Wait, could you say that again?
The other latency reversing agents that are known in literature, could they also be acting through the galectin-9 pathway?
Yeah, so in terms of the galectin-9 pathway, the other latency reversal agents that we've tested are not signaling through the cell surface glycans. And the way we know that is when we actually strip the glycans from the cell surface, it doesn't affect their reactivation potency. However, the downstream effectors are all the same. So we did, for our RNA sequencing from the in vitro experiment suggests that the NF-kappa B pathway and the NFAT pathway are heavily involved in Gal9 reactivation. And so those other latency reversal agents definitely converge with galectin-9 signaling. Once you get past the cell surface, I think that it's a pretty generic reactivation mechanism, I think. Yep.
I'm curious, I think Gal9 is also thought to have a lot of immunosuppressive effects, yes. And if you have concerns about that and what you think about that.
We absolutely do have concerns about that. So the interesting thing, the literature is totally inconsistent on this. So there's some papers there that suggest that it suppresses CD8 responses, for instance, which is bad. We definitely don't want that. And there are other papers that suggest that it boosts CD8 responses or NK-mediated immunity. And kind of what we distilled from the literature that's out there is that it seems to be immunopotentiating in the set of immunosuppression and immunosuppressive in the setting of autoimmunity, which seems maybe like a rose-colored interpretation of the data that's out there, but it's inconsistent. But at the end of the day, I think we can't depend on any of that. So a lot of the work that we're doing right now is actually directly testing in latency models how CD8-mediated killing of the target cell is affected by galectin-9 treatment. And so we have a few different ongoing collaborations to see what exactly happens in this system. But yeah, it's something that we're really, really interested in, because the last thing we want to do is undermine CD8-mediated immunity or NK cell clearance of these cells. Hi.
Yeah, the question is about you showed P21 and Schlafen having a good correlation with this, I mean, this is based on your RNA sequencing data.
Yes.
So if you look at the protein level, from the protein angle, do you see a better correlation with all the other established or reported
good question.
restriction
yeah,
factors?
So what we're doing right now is we're actually using CRISPR to knock out and overexpress P21 and Schlafen-11. The P21 correlation does not seem to be very clear. The Schlafen-11 correlation is very clear. So when we see high levels of Schlafen-11 protein expression, we see very low levels of HIV transcription. And the initial data that we've generated through CRISPR suggests that when we knock out Schlafen-11, that we relieve some of that restriction and we can see more HIV expression. The P21 data in our in vitro experiments so far, although I think there's some issues with the experiments, don't seem to be that conclusive at the protein level.